The humoral response begins with the activation phase, when a cell of the immune system engulfs an antigen. Here, a macrophage engulfs an antigen by phagocytosis. Inside the cell, the new vesicle is called a phagosome. The phagosome fuses with a lysosome, which contains digestive enzymes. The enzymes break down the engulfed particle into fragments in a phenomenon called antigen processing. Within the cell, the processed antigens combine with class II MHC proteins. The complex is displayed on the macrophage's membrane. This display is known as antigen presentation, and macrophages are considered antigen-presenting cells. A helper T cell participates in the next stage of the humoral immune response. This helper T cell has T cell receptors that can bind to both the class II MHC protein and this particular presented antigen. This binding triggers the macrophage to release the cytokine interleukin-1, which activates the helper T cell. The activated helper T cell now releases its own cytokines, which stimulate the cell to proliferate. The cell proliferates to form a clone of helper T cells, all with the same T cell receptors. These receptors are specific for the antigenic determinant of the original processed antigen. The effector phase begins with a B cell. This B cell has membrane-bound IgM receptors that are specific for the same antigen as originally engulfed by the macrophage. An IgM receptor binds to the antigen and the cell engulfs the complex by receptor-mediated endocytosis. The internalized vesicle fuses with a lysosome, which contains digestive enzymes. The enzymes digest the antigen, processing it into fragments. The processed antigen is then attached to class II MHC molecules and displayed on the surface of the B cell. A helper T cell from the clone of cells in the activation phase can now bind to the antigen displayed by the B cell. The T cell receptor specifically recognizes the antigen on the class II MHC protein. Upon binding, the helper T cell releases cytokines that stimulate the B cell to divide and create a clone of identical cells. The resulting B cells develop into either long-lived memory cells or into antibody-secreting plasma cells. Plasma cells have extensive endoplasmic reticulum and numerous ribosomes. Plasma cells are essentially antibody factories. They produce and secrete antibodies of the specificity identical to that of the surface receptors on the parent B cell. Like the surface IgM receptors on the parent B cell, the antibodies secreted by the plasma cells can bind to and inactivate the original antigen.